Dear student, welcome to the course of Basic Genetics, and I am Richa. We are discussing the unit 6 about the gene mapping in the prokaryotes, and in today's class we will be talking about the process, the third process, which is transformation. So how the transformation can be used for the mapping of the bacterial gene will be the focus of today's class, and I hope you have gone through the class material from the last two classes where we discussed about the conjugation and sex junction, and there are some very important concepts. If you have not gone through the lecture 1, I would highly encourage you to first go through that so that you can understand things and it will be much easier for you to follow the concept. So let's first talk about what is transformation, right? So transformation is the uptake and incorporation of a free DNA from the environment by the competent bacteria. What does it mean? So if any genetic material DNA is freely available in the environment, there are some bacteria that can take up this kind of free DNA, right? So this practice of, or this habit of, or this capability of bacterial cell to take up this free DNA is the process of transformation. So it was first observed by Streptococcus pneumoniae by Griffith, and this method can also be used for the gene mapping, although it is much less effective compared to the conjugation. So in this particular case, the DNA enters to the bacterial cell with the help of receptors that are present on the surface of the bacterial cell, and then the DNA binds to those receptors and get its entry into the bacterial cell, and because of that this process also requires energy. So how does it happen? You can see this is the free DNA, these are those receptors I just mentioned, and this is you familiar, this is a bacterial genomic DNA, right? So what is happening? This DNA is going to bind to this receptor here, and then when it goes inside it becomes single stranded, both the strands get separated, and then what happens? Out of the two, remember, one of the strands is degraded, only one strand goes and recombine with the bacterial genome. So it is important because I told you that inside the bacterial cell a linear DNA cannot survive for longer. So if it has to survive and if it has to propagate, definitely it has to have an origin of replication, but it also have to be, if it is linear, it has to be part of the bacterial genome, because then only it can propagate along with the bacterial genome. By itself this small DNA cannot propagate, it cannot, it is just a small piece of DNA without any origin of replication and maybe other things that are required for its survival and to protect it from degradation. So then remember, I am not going to too much detail about these REC protein. These are the recombination proteins, the proteins which are required for the recombination of between the two DNA strands, right, like what is happening here, and they are from A to D, and they are very important for the bacterial cells, right, and in biochemistry you will read about them in much larger detail, but for here for you to, it is important for you to know that for transformation process and for the gene mapping, eventually we need to get this fragment integrated into the bacterial genome, and it happens with the help of recombination proteins. So then it makes the heteroduplex, as you can see here, and then because of this heteroduplex you can see a new strand is getting synthesized when the bacterial cell divide, then the one of the cells will have both the copies of the transformed DNA, as you can see here, and the other strand gets synthesized, and the previous DNA was just like the parental one. So you can see I have highlighted it, one cell is transformed and another is untransformed. So natural versus artificial transformation, which means that, see, as I said, there are several bacteria that can naturally uptake the DNA from the environment. You can see I have listed a number of them like Haemophilus influenza, Bacillus subtilis, Shigella, Streptococcus pneumoniae, even E. coli. They have natural capability of uptaking DNA from environment. However, in the laboratory setup, we can artificially make a bacteria competent to take up this external DNA. So, how we make them competent is a detailed process by which we treat the cells with calcium chloride, magnesium chloride, or we give them electrocompetent, so that if you give them electric shock, they can uptake more and more DNA. So, there are very detailed methods for that, do not need to worry about it. But just remember that the DNA uptake property, free DNA uptake property is natural to many bacteria that are living out there, and we can also introduce it into the bacteria which do not have it if we need it for our experiments. So, purpose of transformation mapping, what exactly it is? We can use this method to test if the genes are linked or not, right. So, linked means, again, see, if it is a piece of DNA which is getting transferred, right. So, anything which is in a close proximity in that piece will be transferred into the bacteria, and so that is why we are calling them linked, right. And if they get a chance to recombine with the genomic DNA, then we can see which genes are getting transferred. So, we can use this method to determine the gene order and also the distance between the genes, and we can also study it to understand the phenotypic differences, right. So, let us take this example where what we are showing, the transformation experiment in case of Bacillus subtilis. So, what it is? We have a bacterial cell which is positive for TYR-A plus and cis C plus, right. So, this bacteria is positive for these two genes, and what we did is we isolated the genomic DNA from these bacterial cells, make them in small pieces, and then transformed it in a bacteria which is TYR minus and CYSC minus. And we want to understand, we do not know, right, this is just for the convenience sake that we have written the position of these two genes, but we really do not know where they are and how close they are and how exactly they are located. So, when you do this kind of transformation, allow these kind of bacteria to grow, and then you grow them on different media plates that are supplemented with different nutrients like tyrosine and cysteine, and then ask under what conditions you are getting individual colonies. That would help you identify the position or the distance between the gene. So, look at this example. So, this part of the genomic DNA, which as I said may have TYR-A plus or CYSC plus, right. There is one possibility that both the genes get the opportunity to recombine, right, with the genomic DNA. So, in that case, when this bacterial cell, which is normally what it is normally TYR A minus and cysteine C minus, right, it will not grow on the minimum media, right. It can only grow on minimum media when it has these two genes. On complete media it will definitely grow, because on complete media anything will grow. So, that is your sort of control, right. And this minimum media, it can only grow when it has both the genes present. It will definitely grow in the minimum media which is supplemented with tyrosine, does not matter for it, and same is true for cysteine. Now, if you come to the other possibility that tyrosine and cysteine, out of the two only tyrosine get this chance to recombine, right. So, if this recombination is happening, you can see on complete media it will be growing fine, on minimum media it is not growing. Why? It is not growing because it is still deficient in cysteine, right. So, if you add tyrosine, it will not grow because it needs cysteine. It can make tyrosine, but it does not have cysteine. It will grow only on the plate which has cysteine on it, right. On the other hand, same example, if the bacterial cell only get chance to recombine the cysteine, the recombination can only happening between cysteine plus and cysteine minus, then it will grow on complete media as all the cells are growing. It will not grow on minimum media because it does not have tyrosine. It will grow now on a media where you have supplemented tyrosine. Why? Because it already has cysteine with it, and you added tyrosine to it, you have given tyrosine to it, so now it is growing on a tyrosine positive plate. And it will not grow again on the cysteine positive plate, as the same argument we have given in the previous condition, that cysteine is something that it can make on its own. It needs tyrosine, which you are not giving, so it is not growing. So by doing these kind of combination analysis with all these kind of combinations, what you can do, you can identify the genes that are recombined, and by knowing their frequency, at what frequency you are getting each class, we will come to an example just in a minute. You can also tell how close and far the genes are. If you have, two are just two, but if you have more than 2 genes, then you will be able to understand and place them in an order with the transformation studies. So I also want you to learn about a very important technique, which is replica plating technique, and which is required for the identification of the trait. I didn't tell you about the method where we were culturing the bacteria in the liquid media and the plate, and I did mention that there are different media nutritional supplement that you can add on the plate. But the biggest thing is that when you have a culture with mixed mutant, right, if you plate it on a minimum plate, then what would happen? If those mutants are not able to synthesize some of the products, then it will not grow. So if something is not growing, how you are going to screen it, right? So this is a challenge. So what you do, you first grow all the mutant or the wild type bacterial set on one plate which has a complete media, right, like here. So on this plate there are all different kind of bacteria will be growing, right, and then you select. So how we select, how we do this kind of selection process is the replica plating. It's a very interesting method. What you do, you make, it's sort of a stem. You make a sterile ball with a stem, you touch it very nicely on this bacterial plate, you just touch it, right, and you touch it, and then what you do, you again touch it to the onto the another plate. Which plate? Which have minimum media without any amino acid. Here in this case we are using methionine, right. So you have one bacterial plate where you have everything but methionine, and this kind of combination you can have for X number of genes, right. You can make a plate with everything except methionine, except tyrosine, except proline, except histidine, so on and so forth. Whatever the bacterial genes or whatever the genes that you are screening for, you can always make the plates with and without them to understand if a bacteria is requiring that particular thing or not, right. So what you did is you have this whole bacterial array and you transferred a copy of it. So you still have this copy, the master copy in a complete media with you. And now you are plating it on the minimum media plate, which is not having one of the amino acid, which is methionine, and then you allow this thing to grow, right. So if you allow these bacteria to grow on this plate, what you will see, there are so many bacteria that are growing here. I just made a circle here. All the bacteria that are present here, they are not growing in this area. It is just a very simplistic way. There could be individual colonies that will not be growing, and then that is what you need to identify. But then which colonies will not be growing here? The colonies that cannot produce methionine will not grow here, right. Only the one that are able to produce methionine will grow here, but if they need methionine, because this plate is not having any methionine, so any bacteria which require methionine will not be able to grow here, and by looking at that you can identify one class of bacteria which require methionine, and same way you can repeat it again and again. So just a closer look, let us see in a better example way how people conduct these kind of experiment. You see, just imagine that this is the bacterial plate, this is a bacterial plate that we have, and we made grid, right, grid on these plate. We mark them as, you know, top, bottom, left, right, so that we can understand which side is what, right. And in this plate, just don't forget that we know these genotypes, just forget. So on the complete media plate, everything is growing, right. And when you put the minimum media, right, you give it a minimum media, then what you are seeing? You are seeing on a minimum media one colony is growing. So there is one colony which is probably the wild type one. It can synthesize everything, and that is why it is able to grow on this plate. Now, if you are adding the tyrosine to the minimum media, just one, so one nutrient. So if you are adding one nutrient like tyrosine to the media, you start seeing that this colony in this grid is also started growing, right. So you identified this colony as a wild type colony. You identify this colony as a colony which looks like definitely deficient for tyrosine, right, because it is not able to grow here, but it is growing in the tyrosine supplemented media plate, right. And now when you add cysteine, you start seeing another colony. So for this colony you can also say that this is something which was cysteine minus, as you can see here, and that is why when you supplemented cysteine, this bacterial colony started growing on this plate, right. So this is a very neat way to identify different mutant types, and why they are putting these two names, because they probably knew that these are the two genes in question that they are trying to understand, and they want to know and separate the bacteria based on which bacteria having what kind of mutation. So this replica plating technique is very important and useful to do these kind of experiments where you have to sort and isolate different type of mutations. So now let us look at this co-transformation frequency. So co-transformation, as the word suggests, it is the two genes that are close together will be transferred together. Remember that. So if they are far apart, there will be rarely co-transformation. So co-transformation, if somebody is telling that frequency of co-transformation high, it means that these two genes are close to each other. So if you see this example that the previous one that we have just did, if you see somebody now add on some data for you to understand the problem more, they say these double transformants. So remember, they might have started with that the recipient bacterial cell might be TYRA minus and CYS minus. So that cell will be minus and will not be growing on the minimum media at all, and then you are doing this complementation assay. So the double transformant in this case are 12. You have single transformant where you get TYRA 31%, and where you get cysteine C are 27%. So if somebody is asking you co-occurrence or co-transfer index, what you would do? You will use the number of double transformants and then divide by the total number, and that is going to give you the index, which is denoted as R, co-transfer index. And so the key assumptions in case of transformation mapping are that the DNA fragments are of limited size, right, 10 to 20 kb. It is very difficult for a bigger DNA to get its way into the bacterial cell. So that is why only limited genes that are in that span can be co-transformed, and high frequency, as I just mentioned, high frequency means the genes are close to each other. And so to summarize, what we learn is the transformation is a powerful gene mapping tool. Co-transformation frequency reveal gene proximity. Gene order is equal to inferred from how often genes are transferred together, and it is very good for analyzing the bacterial gene clusters. Cluster means where the genes are close to each other. So now we will, before we stop, I will help you understand two questions so that it will be easier for you to solve these kind of questions. So let us say if somebody is asking you that transformation, there are two genes that are shown here on the screen, histidine and leucine, and the transformation efficiency of these genes with TRIP. So in a TRIP positive cell, there are 70% of these cell also have histidine with it, and then they have leucine 25%. And if someone asks you the order, then what would you say? You can say that histidine must be close to TRP compared to the leucine, because there are 70% times these two genes were together, right. And now let us take a bigger problem, which has two sections in it. Let us read it. So the three strains of bacteria, each bearing a separate mutation, A minus, B minus or C minus, are source of donor DNA in a transformed experiment. Recipient cells are wild type for those genes, but express the mutation D. So let us say if I want to write the wild type, what I would say? I would say A plus, B plus, C plus, but D minus. This is my wild type. Okay. So now what they are asking based on this? Based on the following data, the data which is given in this table, they are asking that the let us assume that the location of D gene precedes the A, B and C, propose a linkage map for the 4 genes, right. So every cell you see, the donor is D plus and the recipient is D minus, and in every transformant you have D plus. So they are all positive for D, right, but 0.21 frequency they are A plus, with 0.18 frequency they are B plus, with 0.63 frequency they are C plus. So can these frequency help us understand their proximity with D? Yes, we can. So if you see, if D is the first, right, then the highest frequency that it has, it is with the, if we draw it like that, then what we see, the transformation frequency is 0.63. So this is the C. And then what we have the next one is A here. Then what we have the B. So this is our order. This is the order of the gene where they are separated based on the distances that have been, frequency of transformation that has been given here. So next question that they are asking is, if the donor DNA were wild type, right, if the donor DNA were wild type, now they have changed it, and the recipient cell was negative for that, then which of the cross would be expected to produce the greatest number of wild type transformant, right. So the two genes that are closest to each other will give you the maximum number of wild type transformant, and this will be, if you see here, this will be A and B here, right, because these two genes are closest to each other. So with that I stop. I have similar activity question for you on the screen. Please read it and answer the question. And there are actually two questions. One is a very simple one, another is almost the same template that I have shown you to solve here. And with that I stop, and I would like to thank you for your attention. In case you have any question, anything that you want to understand, please feel free to drop me an email or write in the chat box. Thank you. Thank you.